Καλησπέρα σε όλες και όλους που για μια ακόμη φορά είναι μαζί μας για να ακούσουμε άλλη μια σειρά ομιλιών που έχουν σχέση με το θέμα μας της υγείας και της θρησκείας. Η σημερινή παρουσίαση θα γίνει από μία από τις πιο γνωστές διεθνώς ερευνήτριες, την καθηγήτρια Gayle Woloshak, η οποία είναι καθηγήτρια της ακτινολογίας και της ακτινοθεραπείας στο γνωστό Πανεπιστήμιο Northwestern και την Ιατρική Σχολή του Σικάγου. Είναι πολύ γνωστή στους επιστημονικούς κύκλους η καθηγήτρια Woloshak για τη συμβολή της κυρίως σε δύο χώρους, της ακτινοβιολογίας και της νανοτεχνολογίας. Έχει ένα έργο πάρα πολύ σημαντικό, το οποίο όμως θέλει πολλή ώρα για να σας το αναφέρω. Εκείνο όμως το οποίο θα ήθελα να σας τραβήξω την προσοχή και ο λόγος που έχουμε την τιμή σήμερα να την έχουμε μαζί μας είναι για το ερευνητικό αλλά και το εκπαιδευτικό και διδακτικό της έργο σε θέματα αντιμετώπισης του ασθενούς με μια πιο ολιστική φροντίδα. Με άλλα λόγια, έχει συμβάλει με τις ομιλίες της και τις παρουσιάσεις της σε όλο τον κόσμο, ούτως ώστε να έρθουν πιο κοντά οι επαγγελματίες της υγείας, οι ιερείς, όλοι όσοι ασχολούνται με τον ασθενή. Το σημερινό θέμα της είναι η ιατρική τεχνολογία και η Ορθόδοξη Εκκλησία. Professor Woloshak, it's a great honor for us to have you with us today, and we look forward to listen to your presentation on medical technology and the Orthodox Church. Thank you again for your time.
Thank you so much. I'm really happy to be here. Let me get started. Okay, so I'm going to talk to you today about some issues about just selected technologies in the Orthodox Church. The topic is very broad and probably I can give a whole course on it. My email address is here. If anybody has any questions, please feel free to get in touch with me. So I'm dividing this up into several different topics, and I just want to introduce the concept of new technologies. So there have been a large number of new technologies that have come up. I can't cover them all, as I said before. It certainly has become difficult to interface with the rapid pace of technology development. Just in the past few weeks, we've seen the use of artificial intelligence in new ways that we hadn't seen before. I think every week there are new technological advances that take place, and they impact us. They certainly impact medicine. While I'm a researcher, I work in a radiation oncology department, and technology is a major driver for our mechanisms of healthcare. Science impacts humanity in many different ways. Medicine is probably one of the most important. Every person experiences in their life at some point the diagnosis of a disease, the treatment of a disease. We have issues dealing with quality of life. We have issues dealing with end of life. And because of that, ethical considerations really should take place before the technology is employed and not afterwards. Right now in the US, we have ethics considerations for animal studies and also for human studies. But oftentimes that takes place after there has been some determination that this particular technology should be used. I think we need a more broad discussion of technology application as we go along. Now, I want to use one example to explain how very important the science is for the ethics in these situations. And my example is an old one. It's going to be HIV. Many of you will remember the days when nobody knew what HIV was. Some people might not remember it, but before we understood, we knew we had identified AIDS, but we had not identified what the cause was. So in those early days, when we knew that there was this infectious disease that was spreading, that was pretty lethal, in those days, it was considered ethical to talk about quarantining people, preventing people from interacting with others, literally shutting them up in particular environments so they couldn't come in contact with others in order to prevent spread of the disease. But then science did its homework and discovered that the cause of this AIDS was HIV. And now all of a sudden, it was unethical to say we should quarantine people because we knew how we could care for people that were infected with HIV in a way in which it would not infect others. So this is a case where the science drove the ethics to a large example. The facts that we learned shaped how we understood the disease, and then therefore it shaped the ethical treatment of patients. I think you can say very similar things about COVID, although I'm not quite sure we're yet fully out of the COVID situation. But I think as the science learned more and more, how we should behave or how people should respond to that became different and changed with time. So just for a moment, I think most Greeks will know ethikos, the word for ethics comes from the word for custom or habit. It is officially defined as a branch of philosophy. It involves systematizing, defending, and recommending concepts of what is right and wrong, often addressing disputes that are of moral diversity. And I think for those of us that are Orthodox Christians, we probably think of ethics in a kind of narrow focus within a church context. But in most medical practice today, it's a very broad terminology, and it applies to almost everything we do with patients and how we work with them, with particular procedures in place. And that process is to try to protect the patient from having harm done to them inadvertently because of the scientist or the physician who is looking for the best treatment for the patient, but perhaps hasn't thought through all the issues very well. Cyril of Jerusalem, he wrote in 347, I think, something that is worth thinking about. For the method of godliness consists of these two things, pious doctrines and virtuous practice. And neither are the doctrines acceptable to God apart from good works, nor does God accept the works which are not perfected with pious doctrines. For what profit is it to know well the doctrines concerning God and yet to be a vile fornicator? And again, what profit is it to be nobly temperate and an impious blasphemer? A more precious possession, therefore, is the knowledge of doctrines. Also, there is a need of wakeful soul, since there are many that make spoil through philosophy and vain deceit. So what was Cyril trying to say here? He was trying to say that it's great to have these laws. And I would say that even in today's practice with our ethical approach to things, we need to have those. But we also need to have appropriate practices. And those practices are carried out kind of every day by physicians as they do their jobs. Now I've picked on a few new technologies. I hope that I'll say some things that perhaps you haven't heard before. They're not that new in many of these examples. They've been in use for a while, but they will perhaps help us use these almost as examples to reflect upon different perspectives. So the first one is going to be in vitro fertilization, abbreviated in the US as IVF. And there are a series of technologies that sort of surround IVF. In 2002, the US president formed a council called an ethical council, the Council on Bioethics. And they were trying to look and develop an approach toward understanding the possibility of human cloning and understanding human dignity. If you want the full report, you can go to this website and download it. It's a free download. But there was something really interesting about the way that they approached their work. They did not dig into the medicine and the science right away. Instead, what they did is they came up with a reading list of about 20 different books that they chose to help explain who is the human person. But these books were not textbooks. They were actually fiction, meant to tell us something about humanity. What does it mean to be human as understood through our literature, through our understanding of others? And I think that kind of broad approach to ethics is what is needed today. Okay, so let's start out with the IVF. Every person knows this, but let me just make a few, an overview, all the same. So when ovulation takes place, fertilization takes place, the fertilized egg moves up, up, up. This is all of what happens in utero. We have implantation at around day 8 or 9. Perhaps even up to day 10 in some people. And this process is what happens in utero. Now, in probably the past 20 years, there are many people that have tried to deal with fertility disorders. There are clinics that have arisen to deal with fertility disorders by having this process take place in vitro in a test tube instead of in a uterus. Here's just a picture of to put this in mind, and this happens to be a mouse, but it doesn't look all that different. Here's what an egg looks like that's newly fertilized. You can see the nucleus here. You can see the other nucleus getting ready to fuse. Actually, even mammalian eggs have a bit of a shell around the outside to protect them. And now it grows into multi-cell stage. This is now the 16-cell stage, which would, if it were not in a test tube, have probably implanted in the mouse's wall. Humans are really no different. The time frame is perhaps different, but nothing else looks that different. So let me make some points about in vitro fertilization. It is used in the US. The discussion is to treat fertility disorders, most often to help a couple who are infertile to bear children. Although I think in more recent years, it's also been to help women who are single to have children. They were originally called test tube babies. And what do? We isolate eggs from the mother. We isolate sperm from the father. We mix them in a tube, actually most often in a culture dish. We let the embryo develop for a few days. We then implant it into the mother, who has been made to feel like be biologically like she's pregnant using hormones. And of course, there's always the question about what to do with the leftover fertilized eggs. Some people want to use them for embryonic stem cells. Some people believe that these leftover fertilized eggs should be treated as children and should be put up for adoption, or that there should be no leftover fertilized eggs, that the fertilization process in vitro should take place in such a way that there are no leftover eggs. And certainly, in the Orthodox Church, you can find people that stand on all spectrum, on all range of spectra for this. I think one question that people ask is why go to all this trouble of having in vitro fertilization and not adopting? There are many discussions about this. One argument is that there's like a selfish reason why people want to have their own children and not adopt, and that is because they want to pass their genes on to the next generation. But there are other practical reasons. Certainly, the high costs of adoption become problematic. There's difficulty in qualifying for it, and sometimes enculturation issues become difficult. On the other hand, IVF can also be expensive, but these things need to be balanced with each other, and probably every case needs to be taken on a case-by-case basis for what is appropriate. And here's sort of the technology. Here's an egg that is being manipulated. In this example, what's happening is this is being held in place here, and a little bit of material is being taken out so this can be tested for genetic testing. This is in a well case. And again, you can, or multi-cell situation. Here you can see this like little shell around the egg that is difficult to penetrate. You can also use this to inject new DNA into the egg to create some special properties. We'll talk about that a little bit later. There are a lot of limits on doing that sort of technology from an ethics perspective, and I'll talk about some of those. There are some unique aspects that go with in vitro fertilization. First of all, there's pre-implantation genetic diagnosis, where one of the eggs in that small little one of the cells in the 9 to 10 embryo stage can actually have a little bit of DNA taken out of it, and you can test for different kinds of genetic abnormalities. Some people get worried that this will be used for abortion and allow for people to abort the child if the child doesn't have the right genetics. I suppose we can discuss that, but there are other reasons why people do genetic testing pre-implantation. And one of the most important is so that they can be prepared if the child is going to have some sort of abnormalities. So let's say it's a child that has Down syndrome. There are a lot of times when at birth, almost an immediate surgery is needed to be able to correct for heart problems. You can't be prepared for that as well if you haven't done pre-implantation genetic testing. Certainly, there is selection for an embryo that produces particular proteins. There was an example in the US about 10 years ago where a child of two parents had a child that developed leukemia, and they didn't have the right, nobody had the right bone marrow match to be able to help for to cure that child. So they actually had an in vitro fertilized child, but they selected for proteins that would provide the right bone marrow match for the child who had the leukemia. That occurred. One can question the ethics of whether that was appropriate or not, but here we have the sibling who did save the life of his brother because of the in vitro fertilization. Now things have gotten to the point where there is even screening for gender and physical traits. This is an advertisement from a genetic screening place in Los Angeles, where if you want the green-eyed, blonde-haired baby, you can do the selection for the embryo cells that have the right markers, implant those into the womb, and now get the baby of your choice. I think this is a rather selfish approach to doing this, and in the process of choosing these markers, they may be choosing some other things that they don't know about that might be problematic. All the same, this is up for sale and is being used in many locations today. I think we also need to consider the fact that twinning takes place, and this becomes a big issue for many people who are considering whether abortion is appropriate or not, with the idea that twins are two independent people, and at what age can you develop twins, and how soon can you identify that you actually have one versus two babies in utero. The later the separation, the greater the link between the children. So when the separation is at 1 to 3 days at cleavage, you will get twins that have two that will be surrounded by amniotic fluid that's separate, and they'll have two different placentas. If the separation takes place at 4 to 8 days, we will have again two amniotic fluids, but only one placenta. If it goes to 8 to 13 days, we'll have one amniotic fluid and one placenta. And then if it takes place as late as 13 to 15 days, we'll have conjoined twins, which becomes a very different problem. Okay. Now, I'm using that topic to sort of introduce the issue of stem cells. And so stem cell research has become very popular in recent times. It was where people were using undifferentiated cells to replace damaged cells in adults. And the idea was that if there was some function that wasn't working well, then you could just put in new stem cells and have the person work function perfectly fine. And there were certainly diseases with stem cells that people have attempted to treat: diabetes, heart disease, Alzheimer. There are quite a few that have been attempted. And in fact, in the days when people were exploring the use of stem cells, they went after many different kinds of diseases and many different types of approaches, from spinal cord injury, which was very important and is still being treated in some examples, osteoarthritis, much arthritis in the US, both osteo and rheumatoid is being treated with stem cells. I don't think it's been as successful, but certainly it's been attempted. Heart disease, diabetes, etc. Now, I'm going to compare and contrast adult and embryonic stem cells for a moment. So adult stem cells is where we take immature cells from one adult and transplant them into another adult. This has been going on for years. The Orthodox Church has always supported this use. It's the main example that people will know are bone marrow transplants. And in fact, in the US, we frequently have parishes that are doing support for bone marrow transplants by screening large numbers of their population. And now it's been expanded from bone marrow transplant to additional therapies. As I mentioned before, cardiac diseases have been more commonly used. Certainly some spinal cord injuries have been treated. There's some work being done with Parkinson's with stem cells. But the idea in these examples is, if we can't take the adult stem cells and use them directly, we can genetically alter them so that they become programmed to behave like stem cells. So we have adult cells that are behaving like stem cells. The advantage in this system too is that we can actually match for whether there is a compatibility from one person to another. You don't want to transplant cells into a person and have them reject them. The more controversial area has been the use of embryonic stem cells. In general, right now, fetal sources are not used. In vitro fertilization is one of the main sources that is used for embryonic stem cells in the US. In many examples, this is not supported by many of the religious communities. And in the US, there was a very interesting development where particularly the Roman Catholic and a few of the Protestant groups spoke up against the use of IVF-generated embryonic stem cells, said that it was unethical because the IVF-generated embryo is a person, or should be equivalent to a person. And so what they then did is the scientists said this is probably not going to work in the broad sense, and they went and developed these reprogrammed adult cells to be used for stem cells. And this is by far the most common approach for the use of adult stem cells. I will not note that Orthodox haven't really expressed the view. I found varying views in the Orthodox literature. Certainly when I talk to individual bishops, they do not support the extra use of in vitro fertilized embryonic cells. I kind of am of the opinion that we need a lot more discussion on this, because I'm not sure everybody understands the implications of the decisions that are being made. But since most of the embryonic stem cells are not used in today's world, and they don't seem to be making that big a difference to not have them, it really has become, I think, not as broad a question. There are really no applications that are being used today, but the advantage would be that embryonic stem cells can differentiate into all kinds of different cell types, and so therefore they would make a significant difference. And the other problem I think that we have with embryonic stem cells is that we have to worry about transplantation and whether there's a compatibility between the cells that are being transplanted in and those that aren't. But the concept here would be that we have in vitro fertilization in most examples. We now have these cells that are developing inside, that are fetal cells that are developing in vitro in this cells in culture. We pull the cells out and allow them to differentiate into cells for the circulatory system, for the nervous system, for the immune system. And since these are not very mature, the idea is that they could help to develop many different cell types. Now, I've already mentioned this in vitro fertilization, but here's the thing I want to emphasize here is that people have thought about using the leftover fertilized eggs for embryonic stem cells rather than to discard them. I think, as I said before, I think the Orthodox Church in most examples would say, don't discard them, use them for, to allow for more, to implant them into mothers to give rise to babies. As I've noted, I think that this issue has a lot of implications and really needs a lot more discussion than we've had on it broadly in the Orthodox Church. I know there's been some dialogue on it in Greece. So most people that favor stem cell research do not support the use of aborted fetuses for this purpose. And they do not support stem cell research. They don't accept IVF using the extra fertilized eggs. So there is this kind of link that's going on, and this has become, in the US, extremely controversial. Now, we do have these, what are called induced pluripotent stem cells that are generated from adults. These are not coming from embryos. And as I said before, this entire technology was developed to prevent this sort of problem. So the concern about using embryonic cells from an embryo for stem cells was overcome by having these so-called induced pluripotent stem cells. What are these? This is where we might put in a virus into the cells to have them behave in a certain way so that we can generate them and put them into, so they become embryonic stem cell-like colonies that could be implanted into a host of some sort. Now, there are some pros and cons here too. Certainly, the cons, the difficulties are that the stem cells from adults need to be reprogrammed. This is a very low efficiency event. You need to insert genomic DNA that comes with some risks. We'll talk about some approaches now that are reducing these risks. One of the other things that we've noted is that in some people, you inject these stem cells and they become almost like little tumors that grow in the person. And then those are very difficult to treat. And sometimes there's an incomplete reprogramming that occurs, and that means that you don't get all of the cells to behave like stem cells. The advantages are you don't need embryonic stem cells, you don't need to worry about graft rejection. So this gene editing technology that we're going to talk about next or soon I think is one of those technologies that probably will help mitigate some of those risks. I don't know if it will mitigate all of them. And so here's just an example of an animal model where they were using stem cells to create new heart that would actually help to solve problems from heart attack and ischemic heart injury. Okay, so as promised, we're going to talk about gene editing technologies, and this is a very rapidly moving area. My own lab does gene editing in mice. We do not do it in people. We do it in some human cells. So I think there are a number of questions that need to be thought about. The first one is, should we do gene editing? And my answer is we already are. Should we do it in humans? And I'll say we already are. Should we do it in germ cells? I don't know. I have mixed feelings about that, and I'll present that and why. What do we use to justify it, and what influences do we need to consider when we're considering it? So already in non-human species, we've been doing gene editing for probably 20, 25 years. We've done it in lab animals to try to answer questions about how do genes work. How does a particular gene turn on and turn off in a given tissue? Why is albumin made in liver cells and not in brain cells? Trying to understand how the body functions, doing that in animals, and then applying that to what we learn in humans. There are also examples that are used for production materials where we make recombinant products. So for instance, human insulin, for a long time, we only had available to for people that are diabetic to use pig insulin. And what would happen eventually is the person would generate a pretty hefty immune response against the pig insulin. We then started making our own human insulin in cow. And so what we do is put a small piece of a gene fragment that gets turned on in mammary glands, put this onto the insulin gene, hook these two up, put it into the cow's egg. It would then get fertilized, and we'd have a cow grow up that was making insulin in its milk. We milk the cow, we get the protein, and there we go. So this has been going on. Many of you have probably used recombinant products in the clinic. You'll hear about recombinant IL-2 or recombinant IL-1, again, being made from genetic engineering techniques. In agriculture, we create organisms that are more resistant to drought and particular stresses. A lot of people in a lot of countries in Europe in particular do not allow for the use of genetically modified crops. We certainly do quite a bit in the US. And the problem, of course, is you work with a genetically modified crop and the wind takes the seeds wherever it will, and it can spread very easily. Here's just an example of one of the first genetically modified animals. It was a mouse. They made it to express a fluorescent protein, which glows in the dark, in these outside two animals, but not in the middle animal. And you can see how very effective they were at generating this. Now, the second question is, should we do gene editing in humans? And the answer to that is also, we already do it. There are some treatments that are approved for use in somatic cells. They are mostly for diseases that are single gene deficiency diseases. So they involve a single gene. You put in the correct gene and you correct for the problem. Most of our diseases, of course, are not single gene diseases, and there have not been big attempts yet to try to modify those. But most techniques have involved replacement of a gene rather than modification of a gene. With this new technology I'll talk about in a moment, the CRISPR technology, that has that is changing, and I think we're going to see more gene modification popping up. Here's the example. In 1970s, when a child was born with severe combined immunodeficiency, most often this was an X-linked disorder expressed in boys. The solution to their problem with severe combined immunodeficiency was they could not make proper immunoglobulin or T cell receptor proteins, so therefore they couldn't mount an immune response. And so whenever they would come in contact with anybody or anything, they would just die. And so the temporary solution for a number of years was to put them in these bubbles and have really no human touch. It was very hard for these kids. What people did is, when they found the genetic defect, they created viruses that could infect the cells. So they would actually take lymphocytes out of the child. They'd infect them with a virus that corrected the disorder, that had the proper gene in it. They'd put them back. Now the child could live a normal life. Eventually, the virus would poop out. They'd have to go back in after, say, 10 years, reinfect, do this process like every 10 years, but it allowed for these children to grow up, become adults, live in the real world, it solved the problem. But this was an example of a single gene disease and was a single gene correction. So now let's move on to the next question, which is in my opinion, much more complicated. It's about, should we do this in germ cells? So should we modify eggs and sperm and then allow for it to correct for defects. Because many of the defects that we see in people, or I guess I want to call them disorders, many of those disorders are multigenic, for one thing, and they're expressed in more than one cell in the body. So in the previous example I talked about, it was mostly expressed in lymphocytes. You could infect the lymphocytes with the proper gene, correct for the defect. But what about, muscular dystrophy, or, many of these disorders that are expressed in many different cell types in the body? The idea would be to try to develop a technology that you could put into the germ cells to correct for it. My concern about that, and I'll talk about that probably a little bit more a little bit later, and I've written some articles about it as well, is about damage to the human gene pool. So if we make a cow that has a defect because we introduced this gene in the wrong place, well, we can kill the cow. If we put the human gene in the wrong place, we can't kill the human. We've got to have this be an error-free process. In addition, one can argue that there are advantages to even some genes that seem to be disordered in some ways, and I'll mention that in a moment as well. Certainly, there are ethical concerns with this approach. Now, what has changed in the world is the development of this technology called CRISPR. It is a protein nucleic acid complex. It can be used to cut DNA at very precise locations. So what's the problem with the old technology where people would introduce a gene with a virus? Well, the problem was the virus would infect the cell. It could go anywhere in the DNA. It could insert itself in any place, and as a result, could cause what we call off-target effects. That is, it's not just the gene you care about, but it's other genes that are showing abnormalities. What CRISPR does is, because it can be done precisely, because it can target up to 20 different bases, which is almost a single address in the entire human genome. CRISPR has big advantages over other technologies. Here's just a picture here of the CRISPR protein with a piece of target DNA and a guide RNA inside it, so you can just kind of visualize it a little bit. But the concept is kind of like this. You introduce the CRISPR, you introduce a guide RNA with this caspase 9 protein. These come together in the nucleus. You cut out the bad sequence, you introduce the correct sequence. Now this is made into protein, and the disease is corrected. This process, it is really as easy as mixing a few things in a test tube. We do it all the time. It's a very quick, easy process. Probably, so for somatic cells, it's done all the time. What's the advantage? It has great accuracy. Therefore, we have very few off-target effects. And number two, there's a shutoff switch that you can be able to use. So you do it, it does its job, and then you shut it off. Because it has a 20 base pair recognition, you can think about how often a particular 20 base pair recognition, a particular 20 base pair sequence happens in the genome. That gives it great specificity. And in fact, you can even sequence the person who you want to introduce this to, find out whether this particular 20 bases that you plan to use is going to be present in their DNA in any other location, predict whether there are going to be any off-target effects, and then modify your DNA so it's not going to target any other sequences. So this method is remarkable. It uses the cellular typical repair processes to be able to correct for this, and it can correct for a 1 base pair or 2 base pairs. But the idea is it's targeting a unique sequence in the human genome. CRISPR-Cas9, or also there's another one called CRISPR-Cas12a. Those are the most efficient, most rapid most flexible and the cheapest techniques. There are some proteins called nicases that we can use that will minimize off-target effects. And then there are some structures that have been identified that will have slightly different properties so that we can modify them in different ways. But I want to bring up my worries about what to do about doing this with eggs and sperm and the concerns about it. So my worry is about the human gene pool as a whole. So what is the gene pool? It's the combination of all the genes that are present in a reproducing population of a species. So for humans, this would be the collection of all human DNA from a reproductive population. Usually, having a large gene pool is good. Having extensive genomic diversity is good. Inbreeding makes for a smaller gene pool. Correcting for defects may make for a smaller gene pool. And a smaller gene pool makes for species have to have difficulties adapting and surviving. So for instance, you look at the tiger population. They've done sequencing of tigers and they find out that almost all the tigers are very closely related. There's not much diversity there. And the problem is, should there be an infection that hits the tiger population, there's not enough diversity for them to fight this off, and they're going to die. It is the DNA that is in the germ cells and not in the somatic cells. Now, does the composition of the gene pool change over time? The makeup does change very naturally over time through the typical process of evolution. There's genetic drift. There is just, some populations are even today breeding more commonly, having babies more commonly than others are. And so that changes the percentages of genes that we find in the gene pool. Our past gene pool contributes to today's, and today's contributes to tomorrow's, and they are influenced by each other. And there are changes in gene frequencies that will take place. And while you can argue that that a person has influence on the genetic manipulation of their somatic cells die with them. But the change in the cells in the germ line, when you affect eggs and sperm, that gets passed on to the next generation, as long as that person is reproductively appropriate. So I think that there's some hubris associated with claiming we can predict what the impact of mucking around with the human genome is going to have. For many genes, we only know a very small part of what their real function is. And this changes as more scientific discovery takes place. So for instance, I work on a gene, proliferating cell nuclear antigen, PCNA. 10 years ago, it was thought to only be involved in replication. We now know that it has functions in DNA repair, in cell cycle regulation, in many different functions. And as we learn more, we find out that these genes create proteins that are functionally very, very different and unpredictable. So now imagine that you go in and you change a particular gene. Are you affecting other downstream genes as well, or their gene products, the proteins as well? And I think it's very hard to predict. And I'm going to mention in my last few minutes an example that is, I think, important to think about. Sickle cell anemia is an old disease. It's been with us for a very long time, and we do have some good methods for being able to treat it today. But the idea was that if you looked at the sickle cell distribution and the malaria distribution, these were actually very similar, and it was thought that there was a relationship. Sickle cell anemia, if you have two mutant alleles, two mutant genes, you'll die of sickle cell anemia. But in malaria regions, if you have two normal copies, you often die of malaria. Having one normal and one sickle cell copy helps people to be able to survive malaria. So therefore, there was an advantage to having this gene in the human population to help us survive, for example, in Africa and areas of the Mediterranean, even in Greece, without in order to survive. And without the sickle cell gene, humans may not have survived to get out of these situations. So now let's think about what happens if we start messing around with these genes. We might be having influences we don't know. So BRCA1 mutations are associated with increased fertility. So now take out all the BRCA1 mutations. What are we doing to fertility? Alzheimer's genes seem to protect against kidney damage. The ACE genes are associated with long age, and but they also have a relationship to heart disease. So I don't think we can easily say this is a bad gene and this is a good gene, and we want to wipe out this one and we want to keep that one because they have these differences in context. So, you know, if we think about it, genes can be good or bad depending on when they're active, the sex of the person, the life history of the person. For multigenic traits, it's even harder to figure out how particular diseases arose. And then we know that there are contributing factors from infection. And then another issue is that some things we consider to be disease may actually be variations of normal. So I'm going to make my final couple of points right now. There is this desire to eliminate human suffering. That's part of what we do and what physicians in particular care about. Imagine that we can eliminate Tay-Sachs or Huntington's disease. If we do have the ability to cure them and we don't use our energy to do it, then what do we think about who we are as people? Some suffering is needed and even embraced. And then there's the idea of economics, which is treating most of these diseases far more expensive than a single gene editing technique. I mean, the long-term care of a person with Huntington's or Tay-Sachs can be very, very high. It might be actually cheaper to correct it than it is otherwise. I want to just show this guy who he's a postdoc I happen to know, and he generated a do-it-yourself CRISPR cloning kit, which you can purchase online and use it. I don't know how many people feel like doing it. And then here are my kind of final reflections. We have these institutional review boards that safeguard patients and protect them. But I don't know whose job it is to protect against the human gene pool, to protect the human gene pool. IRBs think about the patient, but not about the whole population. Who thinks about the whole population? How do we balance whether this risk is something that we should consider? And I would argue that if the Orthodox Church had a position, what would it be? I don't think it's just like if it's conservative, that's what we should do. I think this needs to be argued. And so I'm going to argue that more discussion is needed. And with that, I'm going to end, and I'll be happy to take any questions or comments.
Thank you, Professor Woloshak, for such a fascinating kind of story of science, of modern science that creates more questions than answers. So if you allow me, I can translate some of the Greek questions. So the first one is as follows. Is a fertilized egg, comma, a 2-month-old embryo, comma, or the delivered baby taking his first breath a human person? Catholics say this is at the conception. Jews say at 3 months of pregnancy or at delivery. What is the Orthodox view? Because many women considering abortion for several reasons look for an answer.
Thank you for that question. In my opinion, it's very controversial for Orthodox. You will find many who say that at conception, we have a baby and that's how we deal with it. I can make arguments against that, and the twinning argument is one of those. I mean, do we consider that that the twins share a soul? No, we don't. We actually consider that they each have their own independent souls. And so up until at least the time of separation of twins at 10 days, 13 days of age, I think you could argue that those are they're not human individuals until that point in time. But I'm giving my own opinion here. I'm sure I'll get in trouble for it. But my own opinion is that a lot of the conceptions that were built around Orthodox thinking about birth were done in the early days of the church when there was not a good understanding of what conception was. The belief actually that up until the late 1800s, it was thought that a man produces little babies, inserts those into the woman, and then the woman must carry those babies. And if she doesn't do it well, then she's done a bad job. Okay, we made our canons and all of our thinking based on a wrong science. And the Orthodox Church has not yet jumped up and said, let's look at this again, but with new science, with new eyes. I think this needs broad discussion for Orthodox, and maybe we'll end up at the same place, I don't know, but I certainly would feel better if we've looked at all the science stories and figured out what we believe is appropriate. Now, I'm not saying I think that science should tell the church what to do, but the church needs to be informed by correct science. So I didn't really answer the question, but I did the best I could.
No, I think you gave the perfect answer. And if the late Metropolitan John of Pergamos was amongst your listeners, would fully agree.
Yes. He and I discussed it actually once.
So another difficult question. You talk about gene editing, CRISPR technology. Are we going to be able to produce babies without the interaction of female and male, having an artificial human? Again, what is the Orthodox view on this future theory or reality?
Yeah. Let me just say, first of all, from a science perspective, it's not possible yet. There are many people that have been working for decades on trying to develop, for instance, an artificial womb. Every time we do this in vitro, we can't get much beyond about the 20-cell stage, and then it starts to behave erratically. Almost becomes like a cancer in some ways, which suggests that the womb is providing something that we don't understand. I don't want to say it's never going to happen. I don't want to say that science is never going to be able to develop the artificial womb. I don't want it to happen necessarily, but it may happen. And I believe the Orthodox Church will, at that time, have to sit through and think through what its stands would be. The things I think the church would have to think about is how much do we recognize, in most contexts, the church says that marriage is the context in which we have children, and that outside of that context, it's not appropriate. The church has not given a blessing for surrogate parents, for instance. And I think that this would be in that sort of format. All that said, I mean, in my own parish, we have, a lot of single mothers, and we wrap our arms around them and hold them tight and take care of their children. We need to be a loving church, and we need to approach everything from love and accept all the people that are coming with all the realities of life. People come at problems, they approach things with different realities. We need to try to understand them and care for them in all those ways. So I don't think the church should be judgmental. I think that we need to be loving and kind about that.
This is just such a great message. And then we go to the last one. You have been an advocate for improved interactions among healthcare professionals, clergy, counselors, and others to provide a more holistic patient care. Can you talk about how you think this could happen? What steps would be needed to initiate this?
Yeah, thank you very much for that question. So I have a slide that I would have talked about had I had time. And that is that biotechnologies are changing the number of decision points people have in their lives. So, think about it. 10 years ago, or maybe 20 years ago, there was the question was, do we adopt children or not? Now the question is, do we have in vitro fertilization? The question now becomes, do we try to select for our babies and what they look like? Do we have a surrogate parent? I mean, these are like a large number of hard decisions. And most often, people don't go to the church for those decisions. They actually go to their doctors for those decisions. And doctors are often not equipped to deal with pastoral issues in that same way. And so what I've been an advocate for is to have teams of people that include like chaplains, pastors, scientists, therapists, all kind of working as a team to try to take care of the patient. And you might have a different team of people depending on the situation you're dealing with. So you're dealing with a family that I had an example of a Jewish couple who were talking about getting an abortion. And it was because there was breast cancer in the family and they didn't want to have the BRCA1 gene transmitted in the family. They were going to have an abortion for every girl that had BRCA1. And you know, this is first of all, it's a misunderstanding of the genetics, but we needed a team to be able to talk with them. It wasn't enough for it to just be one person. So we pulled in a therapist, a psychiatrist, psychologist who could actually figure out what's the dynamic that's going on with them, a genetic counselor to talk about the genetics, the physician who would have been involved in the abortion care. I think all of those together are important. And I think even at the parish level, developing groups and teams like that can be very useful, but we also need it in the clinical setting.
Professor Woloshak, we are again grateful for your today's presence and your wonderful messages. Thank you, and we look forward to meet you in person, hopefully in the future in Greece. Thank you.
So I hope so. Okay, thank you very much.
Take care. Bye-bye. Thank you.